So thank you very much. My name is Nam-Joon Cho. I'm actually working as a material science and engineering department, but I did a gastroenterology, my postdoctoral fellow with the Jeffrey Glenn. It's one of the program with the AViDD and Stanford University. It's project three. One of the topic was targeting viral envelope with antiviral peptide. And then here's our team member, Anneli Varon from the bioengineering, Wachu from the photoscience director, and also Shri Inab from the Scrum Medicine Infectious Disease. So this topic is really combining or a hybrid between engineering and medicine and topic. And then as you all experience from the pandemic, this is one of my interviews during the pandemic era, stocking the shelf for the next pandemic. And the reason I did the interview because there is more to come, and then there is a lot of variants. So we need to have a better solution than now. And a lot of people think, COVID risk has dramatically reduced, but the pandemic is coming. And indeed, a few months ago, or one and a half months ago, in from China and Taiwan and Hong Kong and even Singapore, we had a big outbreak for the COVID again, and then it's an ongoing problem. So in here, the one of the problem is basically there is a lot of unknown, and then as in the material science and chemical engineering and structural biologist, and I first look at the whole pandemic situation in history, and then if you can notice in here, there is two type of virus. One is envelope, the other one is non-envelope. But most of the viral pandemic and epidemic was caused by the membrane envelope virus. So therefore, we need to develop broad spectrum antiviral strategy that work against envelope virus. And then in this particular case, I'm targeting the lipid membrane of those enveloped by the virus. But the problem is the following, because our cell membrane also have a lipid membrane and other parts of organ have a lipid membrane. So you need to have a specific target or specificity. There are, from 1980s, there are a lot of development for the antimicrobial peptide series because they can effectively target the bacteria, either positive gram, negative gram. However, the reason it did not fly because the distinction between the bacteria and cell membrane, it was not clear and then they not effective enough. So in here, the major class of antiviral medicine, and then as you will know, one of the things is we basically demonstrate from 2007, and we name after the lipid envelope antiviral destruction technology, lead technology, which is new antiviral class of a drug that target the lipid envelope of a virus below 160 nanometer, like virus, most of the virus, we can target that. How that was possible? Because we can actually sensing the curvature of the curvature of the lipid envelope of the virus, then we can effectively target. For example, the cell membrane is 10 micrometer. However, the virus membrane is below 150 or 160 nanometer. And then we can actually reading target those curvature, and then we can sense that, we can lysis those things. So, the current antiviral effort to mainly focus on the one virus at a time. If we have those approaches, then you can actually target entire class of lipid envelope virus that belong to those range of the virus. So, mechanism wise, basically we lysis, we read this curvature, you see it here. And then if you think about the 150 nanometer object compared with the 10 micrometer objective, 10 micrometer of the cell membrane is flat compared with 150 nanometer. Then we can actually effectively, those peptide effectively make the tetramer and they effectively read the curvature, sensing those tension to interact and lysis only those have a objective. size below. So, this was the timeline when I did the first, basically identified this experiment, first, basically showing this peptide series in this viral lysis. Then from that is the history of we test with about 10 different virus particles belong to those ranges. And finally, we did a complete study with the Zika virus. It's one of the lipid amylovirus size below around like 100 nanometer or below 100 nanometer. So initial study was we basically accidentally did, and we compare with the two different virus, right, which is one is the Hep C, which is size below uh, in 100 nanometer or 50 nanometer envelope single-strand RNA virus. And as a negative control, we utilizing the vaccinia virus, which is a size shown in here, about 360 nanometer, which does not affect at all for the vaccinia virus. However, it's working against the Hep C virus. So in order to prove this one, we did a lot of study whether the lipid composition of the envelope viruses matter or those curvatures matter. And then later on, we find out through the biophysical and modeling study saying that the efficacy of those peptides interact with those curvature membrane is independent of lipid composition is rather depending on the curvature or tension of the system. It was very hard to argue this one and then persuade the leadership. And then we did about 30 paper of a mechanism study, how this peptide actually lyses conventional lipid envelope of the virus. So one of the main through study was a model system as Zika. And of course, we did the biophysical design of the peptide, which is we increasing the PK of the system and the immunogenesis response developed that. And then also we did the viral neutralization efficacy study with the human cell and the neuronal cell protection and so on and so forth. So this was the actually whole system we actually did for the Zika. First of all, we developed this tethered vesicle platform. The meaning is we can actually coating the lipid envelope with the fluorescence, or inside we can actually encapsulate the fluorescence molecule. And then in here, in this particular case, we basically label the lipid envelope. And then if you see the single dot, that means this is a lipid envelope viral particle. And when the AH peptide interact with the this viral particle, then you can clearly see that upon the interaction and lysis within 10 minutes, you see the losing the intensity of that. And depending on the size of the lipid envelope, you see the correlation of either AHD or AHL peptide, which is the reason we're using the D isomer instead of the L isomer, because we can actually tune the efficacy of the rupturing efficacy of those vesicles depending on the size. With that, we designed this AHD peptide therapy, showed excellent clinical benefit, and then we did a multiple design study, which is first we inject the AH peptide, then we inject the Zika virus with the animal studies. But in this data, in this particular case, we first inject the Zika infection, and then we wait until the day 3, which is the most of the viral particle is promoted. Then we capture the data on day 4 and day 7. You can clearly see that in here, survivor is 80% of the greater 80% mortality reduction. You can actually see it change in body mass. You don't see any of the weird indication intraocular pressure. Also see the reduced eye pressure increase, reduced white blood cell increase. And then with that, we did check for the decrease the viral load in serum, spleen, brain, and optical nerve. In here, you can clearly see that the gray line is basically the viral number, and when you apply the H peptide at day 3, day 4, day 5, you can clearly see the logarithmically you can reduce down in the serum case. Spleen is also you can clearly reduce down very much. And the most surprising thing, number one, the virus is surviving the brain for a long period of time. And then you can see the 2-3 log reduction after you apply the H peptide. And there is no weird sign for the decreasing sign, sign level in the brain too. So that meaning is this peptide can really penetrate the BBB and working against brain nervous system. And then in here, in order to prove that, we check basically how this peptide penetrate the BBB, and H peptide is basically inhibit Zika virus in the brain too. We compare with the dermorphin, which is known molecule of penetrate BBB. We study similar study for the brain influx rate calculation using the healthy mouse, and then demonstrate that indeed this peptide can penetrate the BBB. And then not only penetrate the BBB, effectively when you apply the AH peptide, you can clearly see that you can maintain the integrity of the BBB, intact BBB, basically treatment with H peptide. By without H peptide, you can see the release of the dye, and in here it's to prevent that. With all this, we basically check not only Zika but other type of lipid envelope virus. For example, it's very efficient with the dengue or for serotype, yellow fever, JEV, chikungunya, and so on and so forth. When the pandemic came, so we have designed several different peptides showing that a very potent viral inhibitor for the low nanomolar range, up to 30 nanomolar for the EC50. So for the outcome of this studies, envelope disruption antiviral peptide have a potential to inhibit membrane lipid envelope virus. Ongoing research is going for the SARS-CoV-2 for the envelope virus that causes brain infection too. Rabies is one of our target we study extensively. And this read strategy can be used alone or combination with the other class of antiviral drug and pharmaceutical. So that's what we are basically working on. And then NDA and BLA development process, we are basically entering IND study based on after finishing the AViD this study. So it's a very exciting time to actually move this technology forward. And the one of the thing is basically showing the stereochemistry engineering for the AHD peptide. In here, basically showing the mechanism, and then not only our group defined it, but other group showing that how this peptide can alter. For example, if we have a certain peptide, same class of peptide, but it does not really show any specificity. That means it's no size dependent. But you can actually design peptide 10 actually working against a small size only. And then that part is, as I mentioned, it's very hard to understand, but our group and other group is showing that, and then using our peptide series, they using the neural network curvature sensing peptide system. This is not our group. Basically, they utilizing our class of peptide from our IP. It's they analyze about 50,000 unique sequence from our IP, and then divide by what is the non-binder, what is the sensor, and what is binder. And then they identify that this class of peptide, depending on the membrane binding free energies, is only sensing the curvature and not sensing the flat, like cell membrane surface. So in binder, meaning is antimicrobial peptide series, basically they target both, whether it's curved or have a tension, or have a flat. So using that, our HBVH peptide class, using modification of the capping, showing that this range of curvature sensing peptide, and then the German group is a physics group. They published this paper in Science Advance, correlate with our interpretation of this one. And then in our case, we using, this is not published paper, we using MD simulation interaction between the peptide membrane, showing that depending on tensionless and flat, the meaning is it's mimic the cell membrane, and stretched and curved, that is to mimic the basically viron. You clearly see that in here. Flat surface does not have that much interaction. However, you see that those peptides clearly penetrate depending on the design of the peptide. And in this particular case, the in vitro antiviral characterization AHE dipeptide with the Zika model, you clearly see that cell death we can prevent, even from the 10 nanomolar and 50 nanomolar and 100 nanomolar, you can prevent almost 100% for the cell death. And then it's independent of the different strain of the viral particle. And then I'm showing you again for this data. And in order to actually develop this drug as the one of the candidates, we did a lot of study for the PK/PD study, and in this particular case, we're using the 25 milligram per kilogram for the IV administration, sampling for over 24 hour, high peptide concentration, blood relevant tissue, brain concentration around the 4 to 600 nanomolar. And also the Lawton model half-life is about 6.4 hour, and with the further design with the isomer system. And then dosage with the monkey study, half-life we can increase up to 45.6 hours. So in this particular case, we did it with the collaboration to basically improve our PK/PD system. And then we did the tox study with the mouse and bagel and in a marmoset study up to 60 milligram per kilogram, and see that we don't see any adverse effect for the toxicity study. And then right now, we are moving into the different type of a virus. We're using FTSV inhibition studies with those design series of peptide. So far, we publish and we work on the 27 mer peptide. Now we are working with a 60-mer peptide. That's the shortest peptide possible we can actually design. And then you clearly see the EC50 is a very narrow range of EC50 we can get with the micromolar range. And then the in this particular case, it's showing the in vivo testing peptide with the dengue with the mouse and the marmoset with the system. You can clearly see that the Zika RNA copy number per milliliter showing back to the go back to the background range. So, technology capabilities, we are basically doing with this new mechanism. We are doing with the indication with the Zika, dengue, yellow fever, West Nile, and then also working against with the bioterror threat. And then it is easy to manufacture and store. And then as you clearly see that we basically shorten down our peptide, doing a lot of peptide engineering, and very effectively we can working on. So the one of the benefit and implication for this kind of study is medical countermeasure. You can also using it for the prophylactic, and also we're using it for the treatment study. Thank you very much.